Hello everybody, I'm going to talk about dolichol-linked oligosaccharide diphosphatase and their potential roles in regulating dolichol-linked oligosaccharides. Oligosaccharide phosphates are hydrolyzed from dolichol-linked oligosaccharides, and the oligosaccharide phosphates are seen in increased amounts when the dolichol cycle is blocked, such as in CDG1 or during glucose deprivation. These oligosaccharide phosphates are thought to be generated on the cytoplasmic face of the endoplasmic reticulum. So why are DLOs destroyed in this way? First, DLOs, this is could be a way of recycling dolichol phosphate, which may be useful if there's a block in the dolichol system. The cleavage of dolichol-linked oligosaccharides could be an editing process so that small oligosaccharides which are linked to dolichol are not transferred to protein. Finally, it's possible that dolichol-linked oligosaccharides, if they're unchaperoned for instance, can escape the endoplasmic reticulum and become dangerous for the cell. So our hypothesis is that DLO destruction is required as a constitutive process and required for cellular homeostasis, and it may, for example, be a disease modifier in CDG1. So to test this hypothesis, we have to characterize the OSP generating mechanisms and then use this biochemical information to try and look for functions for oligosaccharide phosphate generating mechanisms. So initially we looked for oligosaccharide phosphate generating mechanisms in crude HepG2 cell membranes, and we found a detergent and cobalt requiring activity. This activity generated from dolichol-linked oligosaccharides generated uniquely oligosaccharide phosphates and dolichol phosphate, which demonstrates that it's a diphosphatase activity. Then we synthesized a series of competitors or inhibitors with differing hydrophobic chains and hydrophilic head groups such as N-acetylglucosamine or chitobiose. And data indicated that the activity prefers hydrophobic diesters. So we call this activity DLO diphosphatase activity. Subcellular fractionation studies indicated that this activity was in the Golgi apparatus and not associated with the endoplasmic reticulum. So how could this activity be involved in producing oligosaccharide phosphates, for example, in CDG1? To test this, we knocked down ALD12 in HepG2 cells to generate a dolichol-linked oligosaccharide with 7 residues of mannose, and also oligosaccharide phosphates, the equivalent oligosaccharide phosphates. Then we treated the cells with bromelain A, and what you can see is that the oligosaccharide phosphates, the MAN7 phosphate, is not altered at all by the bromelain A treatment, but BFA elicits the appearance of a new family of oligosaccharide phosphates generated from mainly mature dolichol-linked oligosaccharide. When we permeabilize the cell membranes of these cells, we find that the truncated oligosaccharide phosphate, as expected, is generated in the cytosol, is found in the cytosolic compartment, whereas the BFA elicited oligosaccharide phosphates are in the membrane-bound compartments or in the Golgi ER fusion compartment. So there seems to be two populations of oligosaccharide phosphates, a new population generated within the endomembrane system, and this occurs while the cytosolic oligosaccharide phosphates caused by downregulation of ALD12 are not affected by the BFA. To conclude, these data demonstrate two OSP generating mechanisms, one on each side of the endomembrane system. The luminal activity would correspond to our Golgi cobalt-dependent activity, whereas the cytoplasmic activity, which manifests itself in CDG or glucose starvation, has yet to be characterized at the biochemical level. So the perspectives are there are two OSP generating systems to be identified at the molecular level, and there is a possibility that the Golgi apparatus may have a role to play in DLO regulation. Thank you.